you should stop the study and not go on. So those are called analyses that are done during the trial. So they call them interim analysis. And this is the only fun part of the analysis of clinical trials, because the other ones are boring, crude analysis, nothing fancy to them. So people, here are some examples that trial that was stopped early for safety reasons, a trial that was stopped by the NIH on the advice of the Data and Safety Monitoring for reasons of futility. Futility is an interesting concept. It means that if I continue towards the end of the study as planned, the results are going to be inconclusive, not positive or negative. It's inconclusive. And inconclusive because you didn't randomize enough people, you haven't recruited enough people. This is a worthless waste of people's time. Because at the end of the day, you're going to say, well, we only had 20% power to answer the question. So you're not going to be able to answer the question. So then why are we continuing to waste money, effort, time, but the lives of the people that are participating in the study? They should be doing something else, right? And so they may stop it by futility. More commonly, that efficacy, the stopping rule for efficacy was met. How do they come up with these stopping rules? And what are interim analysis? So interim analysis are two types. There's the internal periodic monitoring reports that the study team does to assure themselves we're doing the study well, but they are blinded as to who's getting what and what are the treatment effects. The only people that are unblinded to the treatment allocation is the Data Safety Monitoring Board and the statistician that prepares the report for them. So there's usually one person or one group of people that prepares the report, and that person is unblinded to the study results. So just to be careful with the word unblinding or blinded. Blinded, the participant may be blinded to what they're getting. The investigator may, or treating doctor is blinded to what a person is getting. That's the individual blinding thing. But study results being blinded is the entire study. Who knows what is going on in the study on the outcome and on the adverse events is kept from all the investigators, all the study staff, all the patients, only 2 or 3 statisticians and the Data Safety Monitoring Board. And they are looking at study integrity internally, the study team. The DSMB is also looking at study integrity there, but they're the only ones that see the outcomes and events by treatment arm. Okay. Some people say you should never even show them the adverse events, overall adverse events, because they may get unblinded if they know the event rate and they guess and so forth. But that's a controversial issue. So this is the important thing that these people do. So I've mentioned the rationale is that there are multiple reasons why a study could be stopped early. There are statistical reasons because we can analyze this. We can look at unexpected or unanticipated toxicity or side effects, or you've already shown efficacy, or it is zero tendency and unlikely possibility of a conclusive result at the end. That's futility. Or you may be stopped for sloppiness. So you may just not recruit well, so let's just not keep investing more money in you. Because you said you needed 200 people, and it's already been a year and a half or 2 years, and you've only got 5 people recruited? Come on, we don't want to give you any more money. And the sponsor will say, okay, bye, we'll take away your money. Or your data is just so bad, they don't want you, or people are not compliant, or people are dropping out. People are going out and crossing over and doing whatever they want to. In the early days of HIV, there was one study that was stopped. The first study on AZT, luckily was not stopped, but they stopped one clinic because Again, HIV was a death sentence in those days, 1981. And the study at the University of California, San Francisco was looking at AZT. And some chemist put a laboratory outside of the clinic and said, bring me your study drug. I'll tell you if it's placebo or not. And all of these people that enrolled in the study, of course, they paid him a lot of money. They would go and pay money because they wanted to know if it was placebo, and go do something else. And if it was the AZT, they would take it. It spoiled the study. So people will do that to you. If you have a death sentence, you may want to do that yourself, right? But there's other things that people that are very subjective, and that is you may think that you're the only smart person in the world studying this very important problem. There are other smart people in the world. If your problem is important, I'm sure other people are studying it too. So results from other trials may come out. It may be that it's already an approved drug and it's being used in the market for something else. And there's bad effects for that. So even if you show it works here, no one's going to use it. Or changes that whatever you're studying, all of a sudden becomes obsolete. So this would happen in the study that I mentioned the first day on the coronary primary prevention trial of cholesterol, where they had to take that sawdust. Halfway through the study, Lipitor pill was developed. Who is going to take that stupid sawdust ever? Nobody. So why do you continue the study? So the investigators argued, we are not testing cholestyramine sawdust. We're testing lowering cholesterol. And so it's the principle that we're testing of lowering cholesterol. Let us continue. And they begged, and so they already had invested, so they said, yeah, continue. But they have another trial where the opposite argument was given. This was the studies of left ventricular dysfunction. This was given enalapril in people that had low ejection fractions. And this was going on in the US and Canada, while in Europe, they were using captopril. And the captopril study results came out during the enalapril study being conducted. And so they said, well, we've already shown the principle that ACE inhibitors work. So why should you continue your study? Investigators again begged, no. We are not testing the principle. We're testing enalapril. And enalapril has to be taken only once a day. Captopril has to be taken twice a day. Please let us go on. So investigators never want to stop their study. But sometimes the board may say, we have to. We already showed it works.
Analytical stage, the second point says that if there is no treatment difference, not showing treatment difference. At this stage, we are not powered enough to see the difference in the treatment, no, sir.
Yep. So that's a good point, that in the interim, you don't have necessarily adequate power because you have powered the study to have adequate power at the end. So that is a very good point, but power is a probability. And you may have low power, but what if you are lucky to have had a big effect? Even with low power, you can be lucky. So power is a probability. You have 80% probability. You can have high power, 80% or 90%, and not be able to claim significance at the end because you have a 10% chance of not. So it's not a certainty. Power is a probability. And so you could get lucky and have a low power and still get a significant effect, very significant. So that's a good point to clarify. All right, so let's look at a hypothetical example. Suppose I'm doing a study, and because ethics says I'm going to do the study over let's say 5 years, and I'm going to look at the study every year, or maybe it is over 2.5 years, and it's every 6 months I'm looking at it, whatever. But I'm going to look at the study 5 times. 1 at the end, but 4 times in the middle. And so we all know the convenient 1.96, all these things are Z. and we do 0.05 because that is the magical number, and we have 0 to 5 on one side, we do two-sided. So the critical value is 1.96. So I could use that critical value at all of those time points, 1.96. So I can do it, stop the study and say it's beneficial, or stop the study and say it's worse, right? So efficacy is shown or efficacy is not shown. And I can use 1.96. So what do? And maybe I do that. I start my study, 6 months go by, the statistician calculates the Z value and presents it to the board. It's between the bounds, we continue. And what does the board say? The board has to give a recommendation, and they give it to the sponsor, but that recommendation also is in writing and the investigators see it. But you don't want to tell them, oh, we had a Z value of 0.8, because that's given them the results. So you don't tell them that. What they tell them is one sentence. The Data Safety Monitoring Board carefully reviewed the report presented to them and does not see any reason why the study should not continue as per protocol. That's it. No more information. So you continue. Study continues, another 6 months go by, and you get, whoa, now it's gone down. No tendency. Should they stop the study for futility? Well, it's early on, they may decide not to. Let's give them a chance. Let's do that. Because maybe it will bounce back up. Maybe it'll go here. Nobody knows the future. They have to guess, just like everybody. So what does the board do? Let's say they don't stop, they allow it to go on. And what do they say? The Data Safety Monitoring Board carefully reviewed the report presented to them, does not see any reason why you should not follow the protocol. So 6 months go by. Now you're over here. If you were the investigator, you would be feeling happy, and you may change your behavior to make it cross the boundary. But the Data Safety Monitoring Board says, well, hasn't crossed the boundary. What do they say? The Data Safety Monitoring Board has reviewed it, and we don't see any reason why. They don't give you any information, right? Say the same thing they've been saying. Now, T4 goes on, 6 months go by, and you've crossed the magic 0.05 critical value of 1.96. If you were the investigator, you would be happy. But what's the problem? Is there a problem? All of you took a basic statistic course. All of you do know about multiple testing, multiple comparison testing. When you test some multiple tests, you have to adjust for doing multiple tests. So your alpha is different for having done multiple tests. So you need to adjust for having done multiple tests. Have you heard of Bonferroni correction? Bonferroni assumes that you have done multiple independent tests. Are these tests independent, one of the other? They're not. Why? Because I'm using all of the data up to here to do this one, all of the data to do this one. So part of the data for this one is the same part data here. What I'm using here is all of the data so far, and this includes the data I use here. And I don't do this one. If this had been over here, I may not even do this. So they're very highly correlated with each other. They're dependent on each other. And so how do you correct for it? This became an issue in the 1960s with the first long duration clinical trials in humans on chronic diseases. Prior to 1950, there were no clinical trials on humans. A few here and there. There were horrible medical experiments done by the Nazis. Those were not trials. There were trials in Tuskegee that were not trials, that were on the Black Americans, let them die of syphilis, so forth. Those are not trials. But when people start studying they control the infectious diseases, and they start seeing more cancers, they start seeing more heart disease. They start doing chronic disease studies and looking at treating it and looking at long-term trials. And they start to have these issues that what if you show a result before the end? You don't have the luxury of waiting till the end. So in that case, what they ended up doing is say, we need to calculate what's the probability. And this maybe 1.96 I cannot use. I have to do something else. So there's a whole history of this. First of all, and I'll go quickly through it. If you do 5 tests, and each at the 5% level, and these are correlated, they're not Bonferroni. Bonferroni would say it would be 25%. But here, because of the correlation is less, it's 14%. But that's unacceptably high. You don't want an alpha that is that big. So how do you control it? So they used statistical theory of what's called sequential testing, because you're testing it in sequence. And you're repeatedly testing at each analysis. And this actually comes about in quality control in manufacturing. I'm producing toasters, and I start looking, how are the toasters? Are they good quality? Well, I have to check this toaster. And if I have enough bad ones, enough good ones, I continue. If I have bad ones, I stop and I do this. So sequential testing comes from quality control. And there were different tests proposed. Again, you see late 1950s. Peter Armitage in the UK and Anderson in the United States came up with this. Wald is a famous statistician that was very influential in World War II. And he had originally considered what's called a sequential probability ratio test, but that was not used for humans. But again, this Armitage, almost 10 years later, 12 years later, came up with what he called repeated significance testing on accumulating data. And he's the one that calculated that 14.2% that I mentioned. And he said, okay, we can do it, but you have to tell me when you're going to do it and how many data points you're going to use. So you have to pre-specify everything that you're going to see. But then later came what's called group sequential testing. And so equal sized groups of patients are analyzed at, again, pre-specified number of times. And what this is, you say I'm going to do K looks, so 5 looks. And at each look, I will have a critical value. At the first look, I'll have one. At the second one, I'll have another one. Third one, fourth one, fifth one, if I'm doing 5. If my Z value is below the critical limit in absolute value, then I continue. And if I am greater than that, then I stop and reject the hypothesis. And so it gave people a criterion and a Z that changes every time I look. It doesn't stay at 1.96. Okay. So, and those of you that are statisticians, this comes from stochastic processes and the Brownian motion, asymptotic approximation to those Zs. So how do I get those boundaries? Different statisticians propose different things. Stuart Pocock proposed, you know, let's do it one way, and that is, if I know I'm going to do K tests, let's just make it simple. Let's just adjust that 1.96 and move it up to account for doing multiple tests. How much do I move it up? It's a complicated way to do it. He calculated it. And he said, if I'm doing 5 tests, instead of 1.96, it's 2.4. That's it. So for him, this is the Pocock boundaries, 2.413, because I'm doing 5 tests. Ah, Z4, now it doesn't cross 2.43. So what does the DSMB say? The DSMB carefully reviewed all the data, and do not see any reason, blah, blah, blah. Same thing, doesn't tell you anything. So then comes what, another 6 months, and you get a value here. According to Pocock, you would have to say this is not significant. because the critical value is 2.4 and you got a 2.1. Now, what happens when this, at the end of the study, the investigator is waiting in his office or her office, biting her nails, wanting to know the result. The statistician is doing the final calculation of Z5 and comes and says, "Madam, we have the results." And she's ready for it. Yeah, we got a 2.1 Z value. Yay! She gets fantastic. It's greater than 1.96. No. Excuse me. Problem is, we were doing interim testing. We have to adjust for the multiple tests. We're using the Pocock boundary, and it is 2.4. Your 2.1 doesn't cross it, so it's not a statistically significant result. What madam or sir would say, forget about it. We just ignore those testing. It's 1.96. We'll send the paper like that. It's not ethical to do. Nobody likes to use Pocock's because Pocock's difference at the end of the study is a lot far away from 1.96. And so this is not used a lot. Then comes Hay-Bittel in the United States and Pito, that same Richard Pito in the UK, that say, you know, we're going to make it very hard to study, to stop early. Make it 3 up here. So at the end, we can use a much smaller one, hopefully that is closer at the last one, closer to 1.96. And so he does that, and it says, if you do 5, it's 1.99, very close to 1.96 at the end. So with Hay-Bittel-Pito, you are at 3, and here, that 1.99, and this is 2.1, hey, we can celebrate. So people like this one. But then people criticized it and said, wait a second, even when we're close to the end, and this was almost, I mean, big from 1.96, we should have been able to stop towards the end. You don't really, this is not helping me stop at the end. So then comes along O'Brien and Fleming in the United States. You see how the years are changing? People are, now this is 1979, things are happening. And they say, we will have a smooth function of the boundary that will be decreasing as the number of looks gets bigger. So we have 5 looks, and the first look, this is now the square root of 5. But the second look, it's 5 divided by 4, and by 2. So that is 2.5. That is divided by 3. That is divided by 4. That is divided by 5. That's 1. And then I get that at the last one. So it's a smooth function that at the end is slightly more than Hay-Bittel-Pito, but it's also close to 1.96. And it looks like that. It offers the possibility of potentially early stopping towards the end. This one would have been significant. And you don't really want to stop very early unless you have an incredible result. Everybody in this group dies and nobody in this group dies. Then you get a Z that's huge. It hardly ever happens, but sometimes it could happen. We stopped the study in 2017 because they got a Z value of 4, which is incredible value. And it was halfway through the study, or no, 75% of the way through the study. So this is what people use. A lot of people use the O'Brien-Fleming because it has that attraction that as you get more data, as you get more information, you have less variability, you trust your data results more because you have more information, you allow the possibility of stopping, and you don't allow it much unless you have an incredible dramatic effect at the beginning of it. And so it's very attractive to use this one. So others have done, students have to get PhDs, and other people have come up with device. Many people don't use it, but Wang and Sciatis came up with another one that's a mixture of the O'Brien-Fleming and the Pocock. And this is how you compare them. So if I had 10 looks, Pocock would be incredibly high, 2.55 all the time. Nobody likes it. A-beta would be 3, and then here's 2.02. So that's close to 1.96. O'Brien-Fleming is impossible to stop early on, but it's now getting close to 1.96 towards the end. So even if you have a very big result of 2.5 or so, you could stop, but it's a little bit more penalty than Hay-Biddle-Pito. And this is somewhere in between Pocock and O'Brien-Fleming, but you pay a penalty at the end. So I haven't seen this used. I see this one used the most by most people. But the problem was that all of these required that you pre-specify the number of looks that you're going to do. And the DSMBs don't work that way. They say, oh, you know, we have that Z4 is close to it. Let's not wait. Let's have another look in a month. Let's do this. Let's do that. We want to do this. We want to do that. So you do it. Then how do you, well, but we didn't pre-specify. No, I don't care if you pre-specify or not. I want to see it. This is for the safety of the people that we're doing this. So to take into account that people will not stick to the exact one, well, this is how what they look like. In 1983, so 4 years later from O'Brien-Fleming, comes out Lan and the Mets. They said alpha, think about your 0.05 alpha as if it was money. Spend it. You're spending it every time you test something. If you test at the 0.025 at one of the interims, now you only have 0.25 left. If you use it all in the first meeting, then you cannot test anymore. You decide, DSMB, how you want to spend it. So if you ask for another test in a week, then we say, okay, when we do that, how much of the alpha you want to spend for the next test? And you say, wait a second, how does the DSMB know this? The DSMB is clinicians, it's statisticians, it's researchers, it's epidemiologists, it's ethicists, it's patient advocates, it's all of these people as a committee, and they all need to understand this. What is this interim analysis and how it's done and how to interpret it and how to understand it. It usually ends up being the job of the statistician to train the physicians on the board on this so they understand enough that they can all make a decision on it. But this land dimension thing is what everybody does nowadays. It relaxed requirements. You don't have to pre-specify the looks. You don't have to have equal size information. And you don't have to pre-specify when you want to do it. And you have spending functions. And this is just, for those of you that are statisticians, how you calculate the spending functions. But it's all available. There's softwares that will calculate it for you. Dr. Demetz is a retired professor of the University of Wisconsin. If you go to his website, he's got a little app. You can calculate things there for free. So it's not a problem. But you need to know what you're doing. So that's what everybody does. O'Brien-Fleming boundaries with Land-Demetz spending function. And you'll see that in the articles that you read. Now, when you're doing multiple testing, there's also an effect on the sample size. It's small on the O'Brien-Fleming. Even if you do 10 looks, you only have to inflate your sample size by 4%. Nobody does Pocock because you'd have to inflate it 30%. Or this Hay-Biddle-Pito is also small, only 3. So people that are reviewing your grant proposal will ask you sometimes, if you're doing interim analysis, why didn't you adjust on the sample size increase for it? So you better do that inflation. Now, there's other ways of doing it. There's multiple procedures. There's stochastic curtailment. Stochastic curtailment means it's looking at, the conditional power that you have. So given that I've reached this stage, what is the power that I have? The power that I will have at the end, according to my design, is 90%. When I start the study, I have no power. Actually, I have 0.05 because that's my level of significance. You go from 0.05 to your 90% throughout the study as you get more and more data. So you can say, well, I'm here and I have already 85%, then why continue? But if I'm here and I have 40%, then I do want to continue. So that's what Gordon Land had proposed in 82. These people worked on confidence intervals as opposed to tests of hypothesis for that. In 84, these are two British statisticians. And the Bayesian approach was proposed in 1994. All of these things are available, but most of them have not been used. We've used this a little bit, but we also use the O'Brien-Fleming, and in fact, the modification of the Hay-Biddle-Pito as well. So here's an example of it being used. This is the beta blocker heart attack trial that was looking at giving beta blockers to people that had already a heart attack to prevent a second heart attack. Close to 4,000 people. They adopted the O'Brien-Fleming and they crossed the boundary here, close to the end, at 2.82, and they stopped the study early, okay? And when they published the result, it became very controversial. This was in 1981, and the study was going to end in 82. But when they published this, it was in the newspapers because it was a very important study. And 2.8, people started saying, wait a second, a Z value of 2.8. Come on, that's very big. Didn't you have that earlier? Didn't you cross 1.96 earlier? Oh, yeah, we crossed it 2 years before, in May of 79. But we were using the O'Brien-Fleming boundary. And so we did not stop the study until we crossed it. Okay? But people were saying, you know, if I'm the spouse of the person who was getting, I think it was placebo versus a beta blocker, and my husband or my wife died of a heart attack for getting placebo, I would have been really upset. Why didn't you stop the study 2 years ago? My husband had a heart attack last year. And it could have been benefited if they had known the study had been reported earlier. And this is what makes the members of the Data Safety Monitoring Board not sleep at night. They are stressed by worrying, what am I doing by not stopping the study early? Or what am I doing by stopping the study early? Because when you stop the study early, you may not be able to look at long-term effects of that medication. Because once you stop the study, people do whatever they want. They don't follow a protocol anymore. And so you sacrifice long-term effects, or am I sacrificing somebody's health, somebody's life by doing it? It's not an easy decision. Every study is different, but it's important to have it. If you're interested in this study, there's several articles when they announce the early stopping, when they describe their design features, and the statistical aspects of early termination are described very well in that article. Here's another important study that was stopped early. This is the Women's Health Initiative that was looking at postmenopausal women and getting estrogen, and they wanted to see if they could prevent coronary heart disease as the beneficial effect, but also they were worried about causing breast cancer as the primary adverse effect. So they had a primary efficacy and a primary harm. And they used the one-sided boundary for efficacy at the 0.025. They had the one-sided 0.05 level boundaries for harm, and they stopped the study after about 5 years because they crossed the harm one here. And postmenopausal estrogen is not given not recommended for women because of this potential for breast cancer. So these things are used, they change medical practice and so forth. So, yeah. Because they're not interested in harm on the other side. They're not interested in benefit on this side. So they were just, they did one for efficacy, they did one-sided 0.025, which is equivalent to two-sided that, but they are not interested in looking at it. They would not get, if they have something here, they're okay for this, right? But the harm is on this side. You would stop if you have the harm that is below this boundary, or you'd stop for efficacy if you had your Z value for efficacy. They're different statistics, the efficacy and what, right? I don't know if that answered. Okay. So when you make a decision to stop or not stop, when do we have the break? Soon. Okay. When you cross the boundary or not cross the boundary, that's the statistical part of it. But you can decide based on your current interim analysis. You can look at the trend in it. Now you're talking about trending statistics or trending p-values. Your consistency with previous interim analysis, consistency with secondary analysis, because the DSMB doesn't want to take this recommendation lightly. It's a very serious consideration. They want to be really confident that what we're seeing is true. And so we'll torture the data incredibly. They will say, oh, we were doing POCOC. Let's do it O'Brien-Fleming. Let's do it this. Let's do it that. We did this. Let's do it in this subgroup. Let's do it in that subgroup. Let's do it per protocol. Let's do it as treated. Let's do it this. Let's do it that. Let's do this statistic. Let's do this other statistic. Let's do it this analysis. Let's adjust. Let's not adjust. They will ask that statistician to spend 24/7 for a few weeks doing ad nauseum, all sorts of things, because they want to be reassured in making their recommendation. And so they do everything we tell you not to do. They will torture the data as much as they can to see if it is consistent results. And notice what I'm saying. It's consistency that they're looking at. They're not even looking at p-values whatsoever. These people know that that's not the thing to look at. They're looking at consistency of the results. They also look at external consistency with other studies. Are we the only study that are finding this? There are other studies out there. Why did they not find what we are finding? And despite intent to treat, they do all of these things. They analyze in all sorts of different groups. They adjust, they do all sorts of things. And they look at all this consistency in subgroups as well. And why? It's because what he said. I'm stopping early. I did not plan my study to have adequate power to stop early. So did I get a random blip? Did I get something that is really problematic? I'm going to have to report this. When I report this, I will have less precise confidence intervals. I will have increased variability. And so the credibility of what I'm reporting as the hazard rate ratio or the relative risk is going to be questioned because I have wider confidence interval. So I better make sure that I'm making the recommendation correctly. Because once I recommend to stop and the study is stopped, I cannot restart the study. So once the study is stopped, it's never restarted, okay? So we don't want to stop at a random high difference, and we don't want to have false positives. And we don't study secondary events or long-term events because we had planned to have enough of those in 2 years or in 3 years, but we won't have them anymore, okay? So when do you do the interim analysis? So those things are thought about by the board members, right? So once you start your study after you get funding, and you start your study when you have end of funding. So sometimes they'll start it at the beginning of recruitment and they'll look at it during follow-up. But what they do here is just looking at the processes of recruitment, are they doing well, et cetera. They may decide, oh, I want to start looking at interim efficacy here or here. They'll decide when they want to start looking at it. And they will tell the statistician, it's your job, next report, give me this. So I will stop there because this is just the operations of how do you make a DSMB. But that's, it's members are different people nominated by the sponsor, no conflict of interest, an odd number of members. And so their role is data monitoring and safety monitoring. So I mentioned this. They monitor data, they monitor study integrity, they monitor safety of individual patients, collective trends, and study outcomes. They do everything. This is the group that is overseeing the study while it's being conducted. And so they're the ones evaluating the equipoise, and they recommend. They never decide anything. They recommend. It's up to the sponsor of the study to decide to stop or not. And in 41 years, I've only had one time that the sponsor did not agree with the recommendation of a board. So how they do this? Well, this is now more technical, operational, but you'll have this here so you can look at it. There's an open report. There's a closed report where, they have different purposes, looking at study progress, looking at outcomes. If the study has reached a conclusive answer, should it proceed, right? And if it'll never reach a conclusive, should it proceed or be stopped? That's the futility part of it. So I will stop there because